Hello, and a very warm welcome to everyone to our today's webinar on Immunogenicity Risk Assessment for Innate Immune Response, Modulating Impurities. I am Anvesha Bhattacharya and I represent Veeda Clinical Research. Veeda Clinical Research Limited, together with its subsidiary, Bioneeds India Private Limited, offers a comprehensive portfolio of clinical, preclinical, and bioanalytical services to support innovator biosimilar and generic drug development programs for our global clients. Together, we serve clients globally in pharmaceutical and biopharmaceutical, agrochemical and industrial chemicals, herbal nutraceuticals, and medical devices. As your facilitator today, I would like to introduce you all to a couple of points as we begin this session. This session will have a presentation in the beginning, followed by a Q&A round. We have enabled our Ask a Question feature. It's right on your screen where you can drop all your questions related to the webinar right there. Now, moving forward, I would like to introduce you all to our speaker and the panelists for today's session. Mr. Chandrasekhar Kodru currently holds a position of leading the Discovery Biology Department within Veeda Clinical Research Biopharma Division, where he oversees its performance and development of new capability. He actively collaborates with key stakeholders to strategize growth areas for the CRO and enhance the portfolio of research services. Prior to his role, he gained experience at companies such as Syngene International Limited, Advanced Therapeutics, and Jubilant Bioexis, and many more. His previous responsibilities included leading in vitro biology for drug discovery projects in collaboration with sponsors and managing in vitro biology for several multiple small molecule research projects. So, without any further delay, I would like to virtually call upon Mr. Chandrasekhar Koduru to begin this session.
Thank you, Anvesha. Right. Warm welcome to everyone. So as Anvesha has already given a very detailed introduction to the topic that we have here to discuss. So today we are going to discuss about the innate immunogenicity response modulating impurities, which are generally referred to as IIRMIs. So these impurities are present in the drug product, which may end up in the drug product as a result of the process or as a result of the synthesis, chemical synthesis, or as aggregates in the drug product. So these impurities have the potential to trigger immune response, potentially resulting in the occurrence of adverse immunoresponses or reactions. The process-related impurities are associated with innate immunogenicity, and peptide-related impurities are associated with MHC binding and subsequent T-cell activation. The impurities can potentially activate the adaptive immune response system as well through different mechanisms and pathways. However, the IIRMIs may have a significant impact, or they may not have impact at all, or they may have very little impact on the efficacy of the drug product. However, the regulatory bodies warrant an investigation into the immunogenicity of these impurities. Some of the most common impurities that are present in the products are flagyellins, FSL1, zymosans, ODN 2006, poly-IC, high molecular weights, low molecular weight, CLOS 75, MDP, ODN 2216, and of course the LPS. Next slide, please. So how do we assess the innate immunogenicity? The innate immunogenicity can be assessed in vitro systems by the detection of inflammatory cytokines. They also activate the C3A complement system. The innate immunogenicity is primarily is assessed using the human PBMC models or a specific target expressing the reporter cell line assays. AD application for a generic peptide largely depends on the impurity profile as compared to that of the peptide of recombinant DNA origin. Innate immunogenicity is mostly non-specific and is broad in nature, unlike the adaptive immunogenicity, which is generated as a result of a specific infection or exposure. And this is usually the antigen-specific immunogenicity. Innate immunogenicity responses are mostly similar, always unlike the adaptive immunogenicity, where second exposure usually leads to a much more stronger response. So, in a nutshell, the innate immunogenicity is regardless of the number of exposures, each response is going to be pretty similar in nature. Here, the activation of the innate immunogenicity leads to the production of cytokines, chemokines, acute phase proteins, and defensins. So these are some of the markers that we try to see in our in vitro systems to assess the immunogenicity. So what does FDA, USFDA say? They're one of their key document, ANDAs for certain highly purified synthetic drug products that refer to listed drugs of rDNA origin. As per the USFDA document, impurities common to RLD and generic, the impurities in generic should be less than or equal to the impurities in the RLD. And if you're specifically talking about new impurities present in the generic, more than 0.5% impurities is generally not considered as acceptable, whereas the new impurities, which are between 0.1% and 0.5%, they warrant identification, characterization, and the burden to justify for the lack of safety and efficacy, uh, including comparative immunogenicity risk tests. So, how do we do the impurity assessment? The two different sources, as I mentioned earlier, process-related impurities, peptide-related impurities. Process-related impurities are the IIRMIs. These are assessed, these can be also assessed using a DPS supplemented with DS lots. Peptide-related impurities are, usually assessed in the in vitro assays. Other methods include orthogonal assays in silico, MHC binding, T cell stimulation potential, in vitro testing of purified impurities. We know that the PAMs, which are basically the pathogen-associated molecular patterns, which are present on the, in this case, the impurities, they exhibit specific patterns on them, which are recognized by the pathogen-associated molecular patterns, or recognized by the PRRs, which are pathogen recognition receptors. The PAMs are recognized by pathogen receptors, and they are expressed in predominantly most of the immune cells, like the dendritic cells, monocytes, neutrophils, macrophages, eosinophils, mast cells, basophils, uh, T cells, NK cells, and so these are the core immune cells which are basically responding to the pathogen presence in the the detection system. There are multiple families of the PRRs there expressed within the immune system. TLRs, toll-like receptors, CLR-C type lectin receptors, RLRs, NLRs. There are multiple varieties of the receptors present in different immune cells. These PRRs may be cell surface receptors or intracellular, or they may be secreted as the soluble ones. So the PRRs, they recognize the presence of the unique fingerprints on the impurities. So these are recognized components which are common to a group. So they're not specific, like in the adaptive immunogenicity responses. They're the core, the common entities on the impurities are recognized by these PRRs. Like I mentioned earlier, there are different PRR types, cell surface PRRs. TLR2, which specifically recognizes bacterial lipopeptides and lipoproteins. TLR4 recognizes the LPS. TLR2 recognizes the bacterial flagellin. And cytosolic PRRs like NOD1, NOD2, they recognize the bacterial peptidoglycans. present on the gram-positive bacteria. RLR, RIG1, they recognize the viral double-stranded RNA. NLRP3s recognize the LPS. Endosomal PRRs like the TLR3 recognizes viral double-stranded RNA. TLR7 and TLR8, they recognize the viral single-stranded RNA. TLR9 recognizes the CpG DNA, which is basically the bacterial unmethylated one. The soluble PRRs, mannose is recognized by the mannose binding lectin, and N-acetyl glucosamine is recognized by the ficolin. So these molecular patterns present on each category of the impurities are recognized by the specific PRRs which recognize them. So what are the different in vitro assay models available for assessing the immunogenicity? We have several of them, starting with the human PBMCs, of course. Here the FDA guidelines say that 22 donors are minimum required. And of course, the donor population should also be representing a broader HLA type diversity. Here, what we measure are the cytokines, which are basically, predominantly the what we call them signature cytokines for each of the PRR ligands. The IL-1 alpha, IL-1 beta, IL-8, MIP-1 alpha, IP-10, IL-6, TNF alpha, IFN gamma, IL-10, MCP-1, MCP-1 beta, MIP-1 beta, MMP-2, and we also measure the CD4 positive and CD8 positive subpopulations of the T cells. These are the surface markers. We use the flow cytometry to measure all these markers and the cytokines. We also have overexpressing cell line platform where some of the pathway-specific interactions can be assessed. The raw blue cells, which are basically commercially available, they are under the influence of the NF-kappa B, and the CEP expression is a result of the induction of the NF-kappa B coming from the impurities. PRR overexpressing ACT293 cell lines are also available. They also are reporter cell lines with CEP-inducible NF-kappa B. DC activation markers are some of the other things that one can look at for assessing the immunogenicity. The markers include CD11C, CD86, and HLA-DR. And the cytokine release in monocyte-derived DC cells also are an option. Either they can be freshly isolated or frozen. Here, the recommended number of donors is about at least 12. There are other cell lines as well, the MM6, where you can measure the IL-6, IL-8 mRNA levels. THP1 cells are also utilized, where you look at the inflammatory cytokines. And cytokine and chemokine estimation on 3D cultures, co-culture of PBMCs and endothelial cells is also another option available for researchers to investigate the immune responses. So here, why do we choose PBMCs? The primary reason is PBMCs represent a diverse pool of different immune cells that covers a wide variety of multiple PRRs. Here, IL-1 alpha, IL-8, MIP-1 alpha, and IP-10, they represent pathways involved in the 4 types of PRRs, TLR, NLR, CLR, and RLR respectively. These are the cytokines we measure in our assays. IL-6, IL-8, IP-10 indicate the presence of immune response modulating impurities triggering cytosolic PRRs. These cytokines, along with the TNF alpha and IFN gamma, are measured along with enumeration of cell types based on the CD markers CD4 and CD8. We also look at the live and dead cells. This is done using this flow cytometry. Multiple PRR overexpressing cell lines are also available, as I just mentioned. The target-specific outcome may be assessed. basically choose the cell line based on the risk associated with your product. That's how the assessment in a overexpressing cell line is done. However, overexpressing cell lines may have limited sensitivity for aggregates. Our experience, the team experience ranges from peptides to other drug products. Salmon calcitonin 3 reporter cell line studies, vasopressin, teriparatide, semaglutide, liraglutide, and linaclotide. Some of the TLR ligands tested, we have tested using these assays are LPS, zymosan, FSL1, CAM3, CSK, flagellan, MDP, CPG, and so on. I'd like to spend a little time on the human PBMC assay that we routinely do here. Here, the donors are selected on the based on the global distribution of HLA allotypes to basically ensure that the data represents global population. As a part of assay development, we optimize the cell density, cytotoxicity, dose response for the ligands, the sensitivity, specificity, and of course, the surface marker enumeration and cytokine expression. Method validation includes linearity assessment, sensitivity, specificity, surface marker enumeration, and cytokine expression, of course. And also we look at the repeatability as a function of robustness. And the sample analysis contains comparison of the test product and the reference listed drugs with the appropriate positive and negative controls. The method development is done with 3 donors. Method validation is done with 2 donors, PBMC donors. However, the sample analysis includes 22 donors. Some of the key challenges are the inherent donor-to-donor variability of the PBMCs. So there is a theoretical possibility of donor-to-donor variability. So that's something that it doesn't disqualify the data, but occasionally we might see small little donor-to-donor variations in the sample analysis phase. To mitigate that, we try to include some of the donors which are part of the method validation and development into the sample analysis phase as well, as an additional check to see whether these responses are similar as part of validation and analysis. So we have a certain amount of data which is generated as a part of these studies, where we try to look at the sensitivity of different ligands in different assays. In the human PBMC assays for LPS, zymosan, MDP, poly-IC, we see a sensitivity of about 1000 picogram per ml, 100 nanogram per ml, 100 nanogram per ml, and 5000 nanogram per ml respectively for IL-1 alpha, MIP-1 alpha, IL-8, and IP10. So that's in the human PBMC assay. Moving to the overexpressing cell lines, we do see a good sensitivity of about 3 microgram per ml to 200 microgram per ml linearity with a sensitivity of about 0.3 microgram per ml for zymosan A in the raw blue cells. And for the LPS in the same system, the sensitivity is about 0.29 nanogram per ml. Similarly, in the HEC blue TLR2 or expressing cell line for FSL1, the sensitivity is 0.06006 nanogram per ml. And same is the case with PAM3 CSK4 in the HEC blue 3 TLR2 cell line. In the HEC blue TLR4 cell line, we have achieved about 0.078 units per ml for the endotoxin standard. So that's all from me. I would like to thank my discovery team for the contributions. And over to you, Anvesha.
Thank you. Thank you, Mr. Chandrasekhar. Now, moving on, we will be proceeding ahead with our discussion where we are going to answer some of the interesting questions. So, my first question to you is why PBMCs should be chosen for the assay?
So as I mentioned earlier, the PBMCs are chosen because they represent a pool of diverse immune signaling mechanism expressing cell lines. Not if I may rephrase, PBMCs represent a diverse pool of cells which are implicated in the immune responses, like the dendritic cells, T cells, monocytes, macrophages, and all these cells. So that's primarily the reason why we choose PBMCs as one of the models. However, it is not the only model. There are multiple models available, but they are target specific. Here, the PBMCs represent a diverse pool of cells.
All right. So my next question is, what concentration of system suitability control to be used to check spike recovery in the assessment of immunogenicity in drug products?
So this is one of the key parameter that we have to establish as a successful development of the methods. The concentration to be selected is the lowest detectable concentration in the assay system. So that's going to be the concentration which we use for system suitability control.
All right. My next question is why there is need to assess signature cytokines in innate immunogenicity assessment.
So we look at the signature cytokines because they are the primarily secreted cytokines as a response to a specific ligand stimulating a specific receptor.
Okay. The next question is how many number of donors to be used to assess immunogenicity in drug products?
So here, this was rather this is still a very difficult recommendation from our point of view. However, the FDA recommends that PBMC-based immunogenicity assessment be done with at least 22 donors. This is a FDA recommendation as per their guidelines.
All right. The next question is why do we need to enumerate CD4 and CD8 positive T cells?
So as a part of our study, we enumerate CD4 and CD8 positive T cells primarily because we want to see the effect of the impurities on the cell number. The end outcome may be an increase or decrease in the CD4 and CD8 positive cells. So we want to be able to, if it is going to happen, we want to be able to identify uh, and have that demonstrated.
Okay. My last question to you is what other methods are available for assessing innate immunogenicity?
Predominantly, we use flow cytometry. That's very quite often used flow cytometry. But however, if you are looking at the overexpressing cell lines, they are commercially available cell lines have SEEP as one of the reporter genes. So it's not just the flow cytometry. You can use absorbance-based method. Flow cytometry is, of course, primary one. The other methods are, you can also assess the immunogenicity using, advanced platforms like the MSD, Luminex, or ELISpot, or even ELISA as well.
All right. Thank you, Mr. Chandrasekhar. Lastly, I, on behalf of Veeda Clinical Research, would like to sincerely thank you all for being here today and taking out the time to be part of this webinar. I would also like to extend our gratitude to the speaker for being such a wonderful presenter and giving us all learnings today. We at Veeda Clinical Research honestly look forward to hosting you again. Till then, take care and stay safe, everyone. Thank you.
Thank you.